Let us take the next one, that is 8th question number 8. Question number 8 is, how does cytokinesis in plant cell differ from that in animal cell? So here we have to talk of cytokinesis in animal cell and plant cell. And how are these two processes different? Cytokinesis is splitting of cytoplasm, but how does cytoplasm split in an animal cell and in a plant cell? In an animal cell, towards the end of telophase, that is when the cytoplasmic constriction appears, towards the end, cytokinesis has already started. That means the cytoplasmic division takes place from outside going inwards. So it is from outside inwards. This is due to pinching in or constriction of plasma membrane. Whereas in case of plant cell, as they have a rigid cell wall outside the plasma membrane, so here there is a cell wall, and because of the cell wall, the plasma membrane does not show any kind of invaginations or foldings. So when these two nuclei have, or nuclear membranes have reappeared, in the middle of the cell, a small plate-like structure is formed. This structure is known as cell plate. This cell plate is made up of fragments of ER and Golgi. And now this cell plate, it grows towards the periphery. So the basic difference is, this is from periphery to inside, and this is from inside out. So this is the basic difference between the cytokinesis that takes place in an animal cell and a plant cell. We are on question number 9 now. Find examples where the 4 daughter cells from meiosis are equal in size and where they are found unequal in size. So here we are talking of question number 9. The 4 daughter cells, when they are formed as a result of meiosis, meiosis 1 and meiosis 2. Where are they same, all 4 cells, and where are they different? All 4 daughter cells are same when sperm formation takes place in spermatogenesis. So in spermatogenesis, one sperm mother cell, it undergoes meiosis 1 to form 2, then these 2 will divide, meiosis 2 to give rise to 4 spermatids, and they will give rise to 4 sperms. All are equal in size. So this is one division where all 4 daughter cells formed as a result of meiosis are equal in size, whereas this is equal in size. Now when do we see unequal? Unequal are seen during oogenesis. Oogenesis is a process of egg formation where one egg mother cell, when it undergoes meiosis 1, it produces 2 daughter cells, same as here, but the 2 daughter cells are of unequal size. One is big, other is small. The bigger one is known as secondary oocyte, and this tiny cell which is formed is known as the polar body. This secondary oocyte, which is a larger cell, and this polar body, they may also undergo meiosis 2. So, this is meiosis 1, meiosis 2. Again, when this large cell, secondary oocyte, undergoes meiosis 2, it again produces one big cell and one tiny cell. So, this big cell which is produced is the egg or ootid, and the smaller cell is again polar body. So, the cells which are produced as a result of oogenesis are unequal in size. So, our two examples where we find equal sized cells as a result of meiosis is spermatogenesis, and unequal sized cells as a result of meiosis is oogenesis. Question number 10. Describe anaphase of meiosis, sorry, not describe, distinguish anaphase of mitosis from anaphase 1 of meiosis. Let us draw these two diagrams. Anaphase of mitosis and anaphase 1 of meiosis, that is meiosis 1. Now, when anaphase is taking place, I am drawing a stage before that. That means I am drawing what has happened in metaphase. Spindle fibers are formed, and chromosomes will arrange on the equatorial plane. This is metaphase. Sorry, this is anaphase of mitosis. So, what has happened in the metaphase of mitosis? Those chromosomes have aligned on the equatorial plane. What type of chromosomes are these? They have undergone interphase, and it is an equational division. So, equational division means the chromosome number or DNA content has been doubled. So, here we will find one chromosome and attached to its centromere a copy. That means the chromosomes which are here, the chromosomes are 2, but they are fused at centromere. So, if this is the centromere, this is one chromosome and its copy. So, during anaphase, when they have to separate, what will happen is splitting of centromere. So, in metaphase of mitosis, splitting of centromere takes place. So, here after this, the next stage would be one chromosome going here and its copy going here. That means this centromere has undergone splitting. In anaphase 1, the two chromosomes which are there, which arrange on the equatorial plane, they are the homologous chromosomes. Each chromosome has its double DNA, but we normally do not draw it. So, there is one thing which is with one chromosome and with this also, but these two chromosomes are homologous. So, this is homologous pair, and when these chromosomes are pulled apart by the spindle fibers, there is no splitting of centromere. So, here there is no splitting of centromeres. This is the basic difference between the anaphase of mitosis and anaphase 1 of meiosis 1. In anaphase of mitosis, the two chromosomes which are separating are original and its duplicated DNA containing chromosome. When these two separate, because they are fused at the centromere, centromere splits. Whereas in case of anaphase 1, the two chromosomes which are on the equatorial plane, they are homologous ones. So when the spindle fibers, they pull them apart during anaphase, the centrioles are separate, so there is no chance of splitting. So this is the difference between the two anaphases, anaphase of mitosis and one of meiosis 1. Question number 11. Question number 11 is list the main differences between mitosis and meiosis. So let us take question number 11. Mitosis and meiosis. We have done this in detail. So now what all differences are we able to recollect or rethink? First, in which type of cells does mitosis take place? Somatic cells. So it takes place in somatic cells. That means all the cells of the body plus the germinal cells, the cells of the reproductive organ, they also undergo mitosis during multiplication phase. So in somatic cells and germinal cells. Whereas meiosis takes place only in germinal cells. Second difference, how many daughter cells are formed as a result of mitosis? 2 daughter cells. As a result of one mitotic division. Whereas in case of meiosis, 4 daughter cells. Are these daughter cells similar to the parent cell? So, the daughter cells identical to parent cells. In which feature are they identical? They have the same number of chromosomes. So, chromosome number is same. Whereas, in case of meiosis, the 4 daughter cells which are formed, they have half the number of chromosomes. So, they are different from the parent cells. Another difference which we find between these two processes, in mitosis, there is no exchange of genetic material. So, no exchange of genetic material takes place in case of mitosis, whereas in case of meiosis, exchange of genetic material takes place. What does this exchange result in? It results in variations. So, here we can add a few more points, like what is the process which helps in genetic exchange? Crossing over. So, we can write here no crossing over. Here in case of meiosis, crossing over takes place. In case of mitosis, there is no genetic variation because exchange has not taken place, no genetic variation. So, because of this, the parent cell and daughter cells are same because they have the same genetic material copied as it is. So, they can be considered as clones of the parent cell because no exchange has taken place. Whereas, in case of meiosis, as crossing over takes place, exchange of genetic material results in variation. So, genetic variation takes place. Plus, we can add some more differences which are very simple ones, that mitosis is completed in simple 2 phases, karyokinesis and cytokinesis, Meiosis is completed in two main parts, meiosis 1 and meiosis 2. Each one has its karyokinesis and cytokinesis, plus the prophase 1 of meiosis 1 also has 5 subphases, whereas in case of prophase of this, there is just early, mid and late prophase. So that could be additional points, but we must always remember that whenever we writing differences, the first differences should be the most important differences, and then to increase the number of differences we can come to some generalized differences also. Like in case of another point, we can write prophase is not divided into subphases, or you can say prophase is single phase, whereas in case of meiosis, prophase 1 has 5 subphases. In case of mitosis, interphase takes place, and then M, that is division phase, there is no interkinesis. In case of meiosis, between meiosis 1 and meiosis 2, there is one more shorter phase of growth which is called interkinesis. So 8th point would be interkinesis between meiosis 1 and 2, whereas here we can write that this interkinesis not seen. So these are the differences between mitosis and meiosis as the major processes of cell division. This was question 11. So now let us take the next question. The next question is the 12th one, and 12th question is, what is the significance of meiosis? Meiosis is the reductional division. It helps in formation of 4 haploid cells. These haploid cells, what do they act as? They act as gametes. So first important point about meiosis is it helps in gamete formations. It helps in gamete formation. Gametes are haploid, whether they are sperms or male gametes in case of plants, or eggs in case of female organisms. So gamete formation. The gametes which are formed are haploid, and in sexual reproduction, fusion of haploid gamete produces the zygote, and unless and until the chromosome number is reduced to half in zygote, the organism will not get its species specific diploid number. So fusion of 2 haploid gametes helps regain the species specific diploid number of chromosomes. If you talk of human beings, in humans the diploid number is 46. How is 46 number retained or achieved in sexual reproduction? Meiosis in males and females helps in formation of gametes, haploid gametes. Sperms have 23 chromosomes, eggs have 23 chromosomes. When they fuse to form a zygote, the zygote has that species specific 46 number, and now the zygote will undergo only mitotic division. So all cells of our body have that same 46 number, which is species specific to human race. So this is another important point which will be considered as significant point of meiosis. When this meiosis is taking place, the cell undergoes meiosis 1, prophase 1 of that, and those 5 subphases. During this, there is crossing over which is seen, and in crossing over, genetic variation takes place. So, exchange of genetic material, exchange of genetic material which leads to variation. Basically, these variations make us different from each other. One couple, husband and wife, suppose they have 3 children. There are some characteristics which are similar to mom, some characters which are similar to dad or father. How are brothers and sisters identical but they are not similar? They may have some features, you may look at them and can say that okay, they are brothers or sisters, but do they look exactly alike? Do they look like clones? They have differences, and those differences are basically due to these variations, and variations help in better adaptation. So, that is like an extension of this variation. So, these are the significant points of meiosis. Question number 12 was this one. Now, 13th question is basically just a discussion question where the students need to discuss or talk about certain things. 13th one, discuss with your teacher about. There are 2 points which are given. First, haploid insects and lower plants where cell division takes place. Haploid insects. Here we will take the example of bees, honeybees. What happens in bees? Bees lay 2 types of eggs. Haploid eggs. When they lay eggs, these eggs can either be fertilized or unfertilized. So, if eggs get fertilized, they become diploid because here the male gamete would also come, and some eggs they remain haploid, unfertilized eggs. So, these are called fertilized eggs and unfertilized eggs. So, there are 2 types of eggs which are produced by these bees, fertilized and unfertilized. Fertilized eggs produce the females. These females would be of different types depending upon what are they fed upon. If these eggs are fed with royal jelly, we would get a queen. If they are fed with bee bread, we would get workers. So, these females, they are of 2 types. It could be a queen or those could be workers. Queen is fertile and workers are sterile females. What about these unfertilized eggs? They are not fertilized, so they are haploid. They develop into males, and these males are known as drones. Formation of an organism without fertilization is known as parthenogenesis. Now, this unfertilized egg, which is haploid, when it is giving rise to a complete organism, unfertilized is single haploid cell. How does it form a complete organism? Some divisions must be taking place. These divisions are all mitotic. That means all the cells of the drone's body, they are going to be exactly same as the egg in the genetic content. So, egg is unfertilized, has half the number of chromosomes. So, all cells of drone would have the same half number of chromosomes. The second point which we need to discuss here is some haploid cells in higher plants where cell division does not take place. Some haploid cells, some other haploid cells, this was like the first point. Now, let us take the second point. Here haploid cells where division is taking place. Now we are talking of haploid cells which do not divide. In higher organisms, the haploid cells are sperms or eggs, that is male or female gametes, and these do not divide. On the contrary, they fuse to form a diploid or structure which is called a zygote, and zygote divides. So in case of haploid cells which we call gametes, there is no division which takes place in higher organism. This was question number 13th. Now let us take the next one, which is question 14. Can there be mitosis without DNA replication in S phase? Question number 14. In S phase, DNA replication takes place. That means the cell which has 2n number of chromosome will make a copy of its DNA and will become 2n more, that is 4n. This is required because in mitosis, when this cell divides, it will give us two cells with same number of chromosomes. So if this replication does not take place, then how would two cells get the same 2n set of chromosomes? So if no DNA replication, in mitosis we cannot get two cells which are identical to the parent cell. So the question here is, can there be mitosis without DNA replication in S phase? Our answer should be no. Reason, in in S phase, DNA replication takes place, and this is important for equal distribution of chromosomes to the daughter cells. Question number 15. Can there be DNA replication without cell division? Now this is reverse of this. In the 14th question they asked us, can there be division without DNA? We said no, because for division DNA has to get doubled. Now in the next one they are asking, can DNA replicate without cell division? That means, suppose this is the cell, it goes through interphase G1, S, G2, and now it has 2n and a copy 4n and no division. It is possible. Then this cell will be called a polyploid cell. One set of chromosome is haploid, two sets of chromosome which we call diploid. If the number of chromosome increases by a set, such cells are known as polyploids. So now here how many sets are there? 4. So it will be called a tetraploid cell. So this is possible. Suppose after this, the cell is given colchicine treatment. Colchicine is one mitotic poison. It would not let the spindle fibers form. No spindle fibers, those chromosomes will not separate, no new nuclei will be formed. So everything is going to remain in one cell without separating into two daughter cells. So we will get polyploid cells. Nowadays, the hybrids or the plant varieties which we are getting, they are mostly polyploids. Like the wheat which we are eating these days is a hexaploid wheat. So the chromosome number or the DNA replication takes place, but not followed by the division. So this is possible, but the reverse is not possible. That means if DNA does not get replicated, division will not take place. But if DNA gets replicated, division might not take place. So it is dependent on certain conditions. 16th question. Analyze the events during every stage of cell cycle and notice how the following parameters change. So here we are required to, 16th question, we are required to analyze events during every stage of cell cycle. So let us quickly draw the cell cycle. G1, S, G2 and M phase. So, analyze the events during every stage of cell cycle and notice how the following parameters, 2 parameters change. They have given us 2 parameters, number of chromosomes represented by N, number of chromosomes per cell. So, now when the cell enters G1 phase, the number of chromosomes here is 2N, it is a diploid cell. What happens in S phase? It makes copy. So, here DNA replication takes place. So, chromosome number remains 2N, but with double DNA content. So, chromosome number is 2N only, but the DNA content is equivalent to 4N. So, DNA content is equivalent to 4N. Then in G2 it remains same 2N or DNA content equivalent to 4N. This is one parameter. And then what will happen in M phase? In prophase it will remain same, metaphase it will remain same, and in anaphase when the chromosomes separate, the 2 parts will get 2N, 2N. So, again that doubling of DNA will be gone, distributed. So, the 2 cells will get just 2 sets of chromosome, that means they will be diploid. Number of DNA content, that is represented by C, DNA content. How much is the DNA content here? Here N is 2N, that is number of chromosome. C, that means DNA content equivalent to 2N. Clear? Here, how much is N? That is in case of S phase. N is equal to how much? Number of chromosomes is 2N, but DNA content is equivalent to 4N. In case of G2, N number of chromosomes 2N, but C, that means genetic content or DNA content is how much? Is equivalent to 4N. So, difference is the chromosome number remains the same, but because of the replication of DNA, the DNA content got doubled. So, chromosome number we will write as 2N, but we will write that the DNA content is doubled. So, this was the last question on our exercise. So, we have discussed each and every question after the chapter which is given in our NCERT book. Go through all the lectures once again carefully and remember the important points which we need to have in our answers in order to score well. So, this completes our cell cycle and cell division. I hope you have understood each and every point. If there is any difficulty, any question, just revise that particular lecture and I am sure you are going to get it.